Gonadotrophin releasing hormone signaling pathway. The function of GnRHR is the regulation of LH and FSH production. This process is started when GnRH binds to the GnRH receptor. This causes the receptive cell to initiate expression of gonadotrophic hormones. Once GnRH binds to the receptor, the exchange of GDP to GTP on the G-alpha protein occurs. After the activation of G-alpha, a further three subunits are produced: G-alpha S, G-alpha I, and G-alpha Q, each of which controls additional pathways. G-alpha S increases the activity of adenylyl cyclase, which promotes the activity of cyclic AMP. Cyclic AMP promotes protein kinase A, which influences transcriptional activity of the cell for production of LH and FSH via CAMP-responsive element binding protein CREB. The activation of CREB is aided by calcium calmodulin dependent protein kinase type 2, which is activated by calcium ions introduced by voltage-gated calcium channels and by inositol triphosphate IP3 from the endoplasmic reticulum. The major role in regulating expression of LH and FSH falls to G-alpha Q, which activates protein kinase C. Phospholipase C-beta causes translocation and partial activation of PKC, and later full activation occurs by action of G-alpha Q. The activation of PKC causes increase in the mitogen-activated protein kinase pathways including MEK, MEK12, and RAF. They further activate the activity of ERK12, P38, and JNK. The result of the activation of PKC is the creation of C-junction, C-phos, as well as activating transcription factor 2 and ETS domain protein ELK1. All these factors induce the expression of gonadotrophic hormones including LH and FSH.